For millennia, humans have cultivated an intimate relationship with flowers. We use them as objects of aesthetic, religious, and cultural value, exchanging them to convey love, grief, and celebration. But to a biologist, these structures serve a highly specific function. Long before a bud even becomes visible on a stem, the plant undergoes a series of internal decisions. Specific hormonal and structural shifts are initiated deep within the plant's tissues, leading to the differentiation and development of the floral primordium. Beneath the petals and the fragrances we prize, a flower operates strictly as a morphological and embryological reproductive machine. This diagram shows the anatomy of a typical flower. When we look past the protective petals, we find a clear physical distinction between the central female stigma and the surrounding male structures. That whirl of male reproductive organs is the Androecium. Each individual unit is a stamen, consisting of a slender stalk called the filament and a bilobed anther. Across nature, there is immense physical variation in the number, scale, and attachment of these organs depending on the species. Yet despite this variety, every one of these simple stalks and lobed anthers functions as an intricate biological factory, producing millions of potential male gametes. This diagram illustrates a transverse section of an anther. Slicing through the lobes reveals a four-sided, tetragonal internal architecture. Located at each corner are the microsporangia, which will eventually become pollen sacs. They are guarded by three outer wall layers, the epidermis, the endothecium, and the middle layers, which protect the interior and eventually help the anther burst open to release pollen. Moving deeper, we reach the innermost wall layer, the tapetum. The tapetum is responsible for nourishing the developing pollen grains. Its cells are highly active, characterized by dense cytoplasm and typically more than one nucleus. These concentric layers exist entirely to protect and fuel the highly sensitive reproductive tissue packed tightly at the very center of the sac. When the anther is young, that central space is occupied by a compact group of homogeneous cells called the Sporogenous Tissue. Each individual cell within this cluster holds the capacity to become a pollen mother cell. As the anther develops, these cells undergo meiotic divisions. This diagram outlines the genetic math of meiosis. The division takes a diploid cell and reduces its genetic material by half, resulting in haploid cells. These newly formed haploid cells are arranged in a cluster of 4, known as a microspore tetrad. As the surrounding anther matures and loses water, these microspores dehydrate, detach from one another, and develop into individual pollen grains. This microscopic division process, Microsporogenesis, produces the precise genetic payload required to continue its lineage. The mature pollen grain acts as the male gametophyte, measuring roughly 25 to 50 micrometers across. This diagram illustrates the grain's protective structure, starting with a hard outer layer called the exine. The exine is made of Sporopollenin, one of the most resistant organic materials known to science. It withstands high temperatures, strong acids, and alkalis, and cannot be degraded by any known enzyme. To allow the genetic payload to eventually exit, there are prominent apertures called germ pores, where the Sporopollenin is completely absent. Inside a thinner, continuous inner wall called the intine sits the payload. A large vegetative cell stores abundant food reserves, while a smaller, spindle-shaped generative cell floats within its cytoplasm. In over 60% of angiosperms, pollen is shed right here, at this two-celled stage. In the remaining species, the generative cell divides once more before release, creating a 3-celled grain. With its rigid exterior and nutrient-dense interior, the pollen grain is a self-sustaining survival vessel engineered to protect fragile DNA in an unpredictable environment. Once released, a biological timer starts. The grain must land on a receptive stigma before losing viability, or it becomes useless for fertilization. The length of this window depends heavily on temperature and humidity. In critical global crops like rice and wheat, pollen loses viability in just 30 minutes. Other plant families maintain viability for months. To preserve agricultural stability, crop breeders mimic seed banks by storing pollen in liquid nitrogen at -196 degrees Celsius, maintaining its viability for years. But this widespread biological dust has a cost. Airborne pollen grains trigger severe allergies and chronic respiratory disorders, like asthma and bronchitis, in millions. For instance, Parthenium, or carrot grass, was introduced to India as a contaminant in imported wheat. It has since become ubiquitous across the country, acting as a major source of pollen allergy. Conversely, because of its rich nutritional profile, this resilient powder is processed into tablets and syrups, consumed widely as food supplements to boost athletic performance. Pollen's immense biological durability makes it a dual force, a critical agricultural resource requiring careful preservation, and an inescapable environmental factor. This animation recaps the pathway we've traced, from the stamen to the four-walled anther, deep into the microsporangium, through the meiotic division of the mother cell, ending at the armored pollen grain. The colors and fragrances that first attract us to a flower are simply the external interface for a relentless microscopic manufacturing engine.